Okay, so we're just giving people a few more minutes.
Yeah, we're going to give everyone on the line 2 more minutes, then we'll get started on everything. If anyone's having any problems seeing the presentation, it's up and being shared right now. We have the front page for it. Let us know, and we'll make sure that it's accessible to everyone else. But we're going to just give people about 1 more minute, and then we'll get rolling.
Hello.
Hello.
Hi, it's Sue here in Harlow, CRL.
Hi, Sue. How are you?
Hi.
Well,
Sorry, I'm a bit late getting in.
No problem. Yeah, we're just giving people a minute or two before we get started.
I can get the main people on it. We should probably get going.
Sounds
good. Medicine. Congratulations. You've won the award and get the chance to take over and lead on from here. So thanks very much for taking a look at all these slides. You put a lot of good effort. In the second half, we get some interesting thoughts that we'd love to get people's feedback on. So without further ado, over to you, Methan.
Okay, so hi everyone, good morning. I want to thank everyone for actually giving me the opportunity to start leading this effort. You know, we can't forget the brilliant work Jeff has done to really bring the project to this point. I really want to, you know, commend him for what he has done so far. So with regard to the agenda for today, we're going to have a brief intro. I mean, since it's my first time, you know, leading this meeting, you know, I thought I'll do a little introduction and have a sort of a 50-foot view of what the project looks like, kind of give the group a little update of where the project is. And then we're going to get some updates from Alex and his team. Then we're hoping to get a update from David Smith. Then Sue would give us an update on their efforts in Charles River. And then we're going to leave at about 11 o'clock. We're going to have some discussion about some new thought processes going forward. So to begin with, I mean, not to belabor the point, summarize one of the slides that Jeff typically uses just to emphasize how challenging a disease DIPG is relative to some of the other tumor types, and that to express that there is a great need for therapy for this indication. Currently, the only form of therapy right now that's approved is radiation therapy. There are no small molecule therapeutics currently. And one of the reasons for that, I mean, DIPG has been, you know, a challenging therapy to go after for a couple of reasons that I have summarized here. One of them is that it's in the brainstem, and so surgical debulking is really a challenge. The fact that the tumor, as described, is diffuse, tend to, you know, create its own challenging in terms of resection. A lot of the biology for DIPG is not clearly understood, and so there's still quite a bit of work to be done. Hence, a group like M4K has been established and an open science concept to actually help drive that. And it's being really underappreciated that the tumor is very heterogeneous. That's actually one of the challenges, and as a result, you know, there's not a lot of primary tissues available. You have different groups trying to gather tissue and trying to better understand, you know, the tumor types. There's lack of good in vitro and in vivo models that we're trying to establish now. But one of the mainstay challenges for DIPG is that a lot of the small molecule drugs that's being developed don't really penetrate the brain. Another point I keep thinking a lot about is, and hopefully later on some of the clinicians can shed a little bit more light on that, but the question about the fact that radiation is the only form of therapy. The question is, prior to radiation, you know, the tumors, the question is, what's the genetic makeup of the tumor prior to radiation, and has the tumor really changed after radiation occurs? So a lot of the cell lines that we are working with right now The cell line derived from transformation after radiation, and so we're actually looking at subradiation type of tumors after going to selection pressure. Or, are they still the same as the original tumor that exists? That's something to think about as we start some of the discussion. Nonetheless, I think there's a huge opportunity to develop CNS penetrant and tolerable agent to treat the disease. Now, the next slide is more of a summary. I mean, Jeff had put some of this together. It was nice for us to really appreciate the signaling pathway for ALK2, the percentage of patients that really have DIPG with the ALK2 mutation. There's more and more evidence that ALK2 mutation is coupled with, this histone mutation. And, we'll talk a little bit about a paper that just got published that reinforced that concept. I want to highlight the fact that, through our discovery efforts here, trying to find better ALK2 tools, we have identified 2009, which is found to be selective. It's orally bioavailable, it's brain penetrant. It's actually an ideal candidate for characterization of the role of ALK2. But one shouldn't forget, we know the tumor, as I mentioned just earlier, is heterogeneous. And so most likely, we have to understand other lesions in the tumor because the therapies for this type of disease most likely would require combination therapies. And I've just outlined in the last bullet point here some of the other lesions that are typically associated with ALK2 mutation and histone mutation. So we'll discuss some of those a little later. But what I wanted to do in the next slide is just to kind of give a snapshot of what are the characteristics of 2009. We think it's a very, very good candidate right now to better characterize the role of ALK2. And so I've just summarized here some of the features of 2009 relative to some of the target profile that we have. And everything is looking really, really good so far for this compound. We are currently waiting on some validation in some of the efficacy models. And so we have the FOP model that's ongoing right now. We'll talk a little bit more about that later. But hopefully later we'll discuss some of the DIPG models that we think compounds like this should go into. In addition to that excellent profile I just showed you, I wanted to emphasize that some of the additional features that 009 has, it's orally bioavailable in rodents. You could see very, very good bioavailability in both rat and mouse. So for any sort of in vivo efficacy studies, I think it's an ideal candidate. And for models that actually demand IP, some models may not find oral route the best route, so the compound can also be dosed IP, and so we get very, very good exposure for any subsequent IP studies we want to do. I think what's also important is we saw very, very nice linearity with dose escalation up to 100 mg/kg. This is exactly what you want to see when you're going into the clinic with compounds of these types. Very, very nice linear escalation with dose. And finally, from the minimal studies that we have done so far, we were happy to see that the compound is very well tolerated up to 100 mg/kg. So you can see with no body weight loss. So we are convinced that if ALK2 is playing any critical role in the disease, 009 is actually a very, very suitable candidate for proof of principle. And so as we define those models going forward, we're hoping that we'll be able to test 009 and really establish whether or not ALK2 alone is enough. The next slide, I really want to commend the efforts of GL Chemtech, who's been on board with the open concept. So thanks a lot, GL Chemtech, for delivering, gram quantities of the material, which we plan to make available to the community to better establish the role of ALK2 in the disease. And so we have about 42 grams right now currently available. Some of it is currently being shipped to us. Okay. On the second to last slide for this section, what I also did too, in addition to 009, we have two potential backup candidates to 009, that is 2117 and 2143. And what I've done alongside 009 in the target product profile table next to, I've tried to establish what is it about these backups that sort of set them apart to 009. And so if you see 2117 and 213, you could see that they have much better selectivity than 009. It's almost twofold, 113-fold over ALK5 for 117, and about 104 for 2143. In addition, we think 2143 also has very, very large volume of distribution. That might be a feature we might capitalize on as we think about getting into tissues like the brain and want to get good distribution into areas like the brainstem. So I think that's a backup feature that we can capitalize on as we establish more and more what role ALK2 is playing in the disease. And so, we are confident we have some nice ALK2 candidates. So if ALK2 really have a role in the disease, I think we have the tools to establish that right now. And on the last slide.
Before you go into the last slide, can you just speak briefly about where we are on the other testing for 2117 and 2143? There's a number of things on test on the games and the SIP innovation.
Yep, so that's something we have to establish. So these dose escalations, so we have to decide whether or not people think that these are candidates ready to go forward. And if the team agrees, then we have to get enough material to do some of those. So like dose escalations that will take a little bit of time. So we have to decide whether or not we want to do that now, or we actually want to do some work on 2009 to establish whether 209 is indeed a candidate. And if it has deficits, then these would be stuff to look at, and then we gather that data. So something we should discuss, whether we get more of that data now, or we actually wait until we evaluate 209 a little bit more. But just wanted to highlight them as potential backup candidates. Okay. And then the last slide is kind of what are some of the critical next steps that we perceive going forward. And if people have other additional thoughts, feel free to chime in. So what I wanted to highlight is that with the lack of any good DIPG models right now, I think the group had decided let's go and use FOP as kind of an on-target proof of concept model. And so that model has been tested in Eileen Shore's lab. And luckily, Owen and I had a discussion with Eileen. We got an update from her, and actually the experiment had started last week. And I think it's supposed to finish the end of this week. She plans to now collect tissue and blood, and then she'll start the analysis. So I think it's, thanks to Eileen for getting this off the ground. Just wanted the team to know that the experiment has been started. So we're about halfway in. Okay, we'll keep you apprised of how things are going. The hope is when this experiment is done, we can get actually Eileen to speak at one of the team meetings to kind of give us an update of the summary of the results and what to do going forward. What I have on this slide is basically some of the details of the experiment that she was running. So it's a typical 5-arm study. Just to remind people, we are doing doses up to 100 mg/kg once a day. We plan to collect plasma at 24 hours. After the last dose, we're going to collect brains. Then she'll do some of the analysis of the bone tissue. Some histology will be done. Ahmed has already provided us with formulation information to get her started. And so after this data set, we'll decide what other experiments we want to do following up. Okay, so that should be exciting. Looking forward to that. The other big thing that's kind of brewing over time, I mean, it's something we're kind of new up front, but it's becoming more and more of a reality, is that the disease therapy definitely might require combination therapy. So we're starting to think long and hard about combination strategies. So the question is, what would be ideal to combine ALK2 with? Now it looks like ALK2 is a key mutation in the initiation of the disease. And so in addition to some of the histone modification, some of the lesions that we are considering, some of the things like PDGFR, receptor amplification, there is some PI3 kinase deregulation, mTOR, CDK4. These are some of the other kinases that might be deregulated in the disease, and we thought these might be good kinases to the combination with ALK2. In addition, people have established the role of epigenetics, particularly the role of the PRC2 complex and EZH2. Now, one of the challenges with some of these EZH2 inhibitors is that they're not very brain penetrant. However, we're hoping that an alternative to that, if they're working, would be the EED inhibitors, which are much more brain permeable, and so those would serve as an alternative. What's also interesting is that groups like Javad and those have established HDAC inhibitors, actually have efficacy in the DIPG lines. Again, however, a lot of these are not very brain penetrant, and so there are 1 or 2 brain penetrant HDAC inhibitors out there. One of them we have in-house, so we're going to probably try and evaluate that. Another thing we want to do going forward is to establish really a lot of the DIPG lines that we have now. The key question is ALT2 in any way driving the growth of some of these lines? And if ALT2 is not driving the growth of these lines, then that could be some of the reason why we're not seeing much efficacy today. And so a bit of work still needs to be done to establish whether the lines that we have in hand are driven by ALT2 in any way, and would hopefully better help us understand how to do some of those combination therapies. And so some of the things we're thinking about, to do some CRISPR knockout, and other strategies to do chemical knockout using PROTAC based on some of the selective ligands we have now. And, we continue to appreciate the efforts by a lot of the other teams trying to develop efficacy models, mouse models, for such a difficult to treat disease. So groups like Chris Jones and, Parker Boser and Jerome, they're all putting significant efforts into establishing ideal models. But lastly, I want to mention that, again, to the team, 009 is now available to the community, so just let us know if you need compounds to do any critical studies. I will have some shipped to you. Okay, so that's basically what I had for the intro. If anybody have any burning questions, I'd be happy to answer them. I'm going to come back later with the second half of the discussion around, some of the thought processes that we're thinking about for the discussion. So next up was Alex, I think. Yeah, Alex did mention that he might.
I have a question about the PK. For the half-life enables the QD rather than BID dosing for the efficacy.
Let's go back to the picture.
Yeah, you were here.
Yeah, so what we're getting at, we have a half-life of about, what, 2 hours in mouse? So, yeah, the exposure. Would you comment on that?
So, sorry, the question is, you are saying that can we do BID instead of...
QD enough, that's what I...
Well, that is hard to answer.
Based on PK.
Yeah, so it's hard to answer right now because we don't know, if we knew a cell-based EC50, for example, right, we can go to a team and take it from there. Right now, we are running in an FOP model, which is a different model than a cell-based model. So it's hard to answer because we don't have all the pieces to answer your question right now. So, and this is why we are just doing a dose escalation with the QD dosing for FOP model. In future, when we know all the pieces together, and model, right model, cell line, and we'll plan it.
Okay. Okay, so Alex, you're next up.
I thought it was Jerome.
Oh, so Jerome, yes, Jerome actually emailed me, sorry about that. He mentioned that something came up and he wouldn't be able to attend today.
Oh, okay.
Yeah.
Okay, so what have we got first? We've got Roz first, and then she'll hand over to John too.
Yes.
Over to you, Roz.
Hello.
Hi.
Hello, everybody. So we've just got a kind of quick update and, I guess, summary of all the structures because, well, anyway, this will be my last M4K meeting. I've moved on to a different group. But just the latest structures that we've got are M4K 3007 and 2112. So these images are just showing 3007 on the left and 2112 on the right. I've kind of removed the beta strand that sort of covers the pockets so we can see properly what's going on in there. The main thing that you can see with the 3007 is that there are still waters in the back of the pocket, which we haven't seen with any of the trimethoxy compounds. I think because the left-hand methoxy group kicks those waters out. So because of the way the molecule is kind of bent round to the right in toward the back of the pocket, it's letting some waters in at the back. The 2112 I got on Monday morning, so it's still just in the early stages of refinement, but it was only about 2.7 angstroms, so We can't see any waters in that one, but the trimethoxy pose is very similar to the others that have that same head group. The solvent side is a little bit... Hello?
Sorry,
Hello?
Rod, this is
Oh, hi, hello. Go ahead.
Hi. Just to let you know that Sylvia, our CAD representative, has been having a look at your crystal structure. We've got a couple of slides in our sections that she'll cover. So,
Oh, that's fabulous. Okay.
Yeah, she's been looking at that.
Okay.
Cool.
Nathan, do you want to change slides?
Yeah, Violet, yeah, okay, let's go.
So this was just an overlay of the 3007 and the 2009, just to have a look at the different poses of the molecules in the pockets there. So you can see with the 3007, it's much more bent sort of forward, and that's why those waters are still available there. So the 2009 has the blue waters, and the 3007 has the red waters.
Thanks.
And then the next several slides are basically just a summary of all the structures that we've got so far. I'm very sorry, I left one off, which was ALK2 with 2044, okay, 2044. That was done much earlier last year. But yeah, so I've just put a summary of the resolution, the compound, which protein it was, and then just a cartoon diagram showing the ligand in the pocket. So we have 2118, 2121. So 2149 and 2184 were the ones with the amide head groups, and I did send 2184 to the synchrotron on Sunday, but sadly those crystals didn't diffract very well at all. And I did have much bigger, better crystals of 2149 because there wasn't, that one's at 2.6, so it, you know, you can see the ligand, but you can't see the waters or anything. But sadly, the bigger 2149 crystals also didn't diffract very nicely. So this is what we have for the moment.
So Sue and Metvin, David, just tell us in future, you know, what M4K compounds are critical to crystallize, and we'll prioritize.
Okay.
And
Sounds like
also, just shout if there are any particular data sets you don't have but you want to see for modeling. And similarly, I guess we're looking at depositing the structures, putting them in the public domain. So if there's any that you still want to look at while we're refining them and doing that, just let us know.
Okay, yeah, sounds good.
We'll move on to John Fu.
Yeah.
Next slide.
Sorry, just to say, before we deposit them, if you could let us know which chemists made which molecules for the structures that we've solved, so that we can put them on the author list. But most of them are in the sort of final stages of refinement, so they should be done reasonably soon.
Okay, just let us know what you need and when you need it.
Absolutely.
Okay.
Okay.
Next slide, please.
Yes.
Yeah. Hi there. I'm John Fu. So I've included like 2 slides from the previous meetings, just as a reference in case if we want to go back and look at some of the data. So maybe we can already move on to the next slide. So these are the previous in vitro efficacy assay where I was checking the viability of the different cell lines, different DIPG lines with different mutation, and as well as the wild type DIPG with the DIPG cells with the wild type ALK2. And on the second previous data slide here is because we were wondering what's the difference between the patented compound, the 3003 and 3007 versus our M4K 2000 series. And in the past, I have done also not only the nanograd, but and the dual luciferase assay, but as well as the phosphorylation of downstream substrate of ALK2 by immunofluorescence staining in the cells, and they are quite comparably active, although So,
Does that result need repeating? Independent replicate? No.
Yeah,
Technical repeat.
yeah.
Okay.
So I had to technically repeat, like,
Okay.
yeah. Actually, there are plenty of things that I should, like, repeat. But, yeah. So, we can move on to the next slide.
Okay. So one quick question. So you think a lot of that efficacy that you're seeing is alpha-driven, or it might be something else? With 303. You think that? And whether or not that's what? Sorry?
I think the connection wasn't.
Saying, is this ALK2 driven for the 3003 on the DIPG 21? And I guess you can't definitively say that.
Yeah.
The molecule is partly uncharacterized and partly you're hitting 4 or 5 kinases.
Yeah, because the 3003 it is also like hitting some off-target kinases as well. So.
So do we know that? Have we profiled 303? Or is that something we should do to understand that biology?
I don't think we've done it.
I don't remember seeing any other off-target kinase panel on that compound.
Maybe we were discussing whether or not, I think Alex did mention about some profiling for 007, and I'm wondering if that should be the one that should be profiled instead. I mean, it's just something to discuss as we try and understand what we. Sorry.
I think we should do them both.
Yeah, because.
Yeah, I think we should do them both just to understand the profile. Now, you might not have enough of 3007 because I know we were getting very, very short of material, but we have intermediate, so we should be able to make some more if necessary.
Yeah, very much.
So just.
This is Al. So if we think of it logically, right, we were trying to make a molecule that is an on-target in the cell and in vitro, and we've got it. And then we have everybody freaking out over the lack of efficacy in these weird-ass cell lines that, honestly, we have no clue if they have any relevance to reality at all. And now you're looking at one data point from a weird-ass cell line and talking about which profile. I am literally ignoring the cell line data for now because we could go sideways into a million rabbit holes worrying about this. I mean, we have a genetic dominant mutation in a person that every tumor has of this subset. The hypothesis can only be tested in a person. Every time we worry about these weird-ass cell lines, we're distracting ourselves, in my opinion. You've got awesome data for the nanobreadth, the kinase activity correlates with that. We are hitting the target. And so I would fast forward into the clinic as fast as we can.
And what are you basing your combination other therapy on?
Don't worry about combinations for now.
So, we've asked some clinicians to join the call, et cetera, because this could be a good conversation with them. Yeah. And, there should be, if you give a patient this drug, there's a good chance they're not going to be allowed on another drug. So we have to make sure that we think for these patients, we're giving them the best choice. So where we're going with all this is, in my view, is trying to understand what if ALK2 is not working on its own as a hypothesis,
where is that hypothesis coming from? Just the H3.3 mutation as well.
The cell lines.
No, that should not factor into it at all. The genetics is clear. We have a 3.3 mutation, and so it's reasonable to assume that it's a two-thing process, and we will need. But I tell you, every neuron wasted on this.
To the best. Boy, we're talking about the ultimate size of scientific knowledge here. But, to think we need to have some kind of basis, I believe, and I'm curious to find out more about what are the two best highly likely combinations. And that's along the lines of, I think, what we're trying to understand a little bit here and a little bit of the discussion that's coming up here, so we can pursue what we think is the highest and best chances of the combination.
So that's what I said. I mean, the team has, it's up to the team to
Throw that
decide.
block in the pond and see the splash.
So those are some of the discussions we're having. I mean, it's up to the team. So we know there's an ALK mutation in the disease. Okay. Apparently, as we get more and more evidence, that ALK2 in combination with some of the histone modification seems to be critical to driving the growth of the tumor. Now the onus is on us. Are we willing to take the development risk, like Al said, and take a selective compound that hits the target and go and prove clinically whether or not ALK2 is valid? So, I mean, that's a valid point. The other option would be, one, we have some compounds that are actually active across some of these lines that have ALK2 in addition to other lesions. So, you know, I mean, those could serve as backups, or if you feel like they're efficacious enough in some of these cell lines,
ignore the cell line.
Yep. Okay.
So this is unprecedented, right? I mean, we're doing something that nobody else has ever done.
Yeah.
I mean, as long as we can all agree that this is something that's completely unprecedented.
Yes.
If there's precedent, then
Very
that's
few drug discovery programs are driven by dominant mutations in a gene. Remember, the world of genetics now is superseding what we all used to do before: upregulation, downregulation. We used to have to construct stories as to why the therapeutic hypothesis was right.
Mm-hmm.
In this instance, the genetics is clear as clear can be, right? And so you have to only look at the subset of genetic experiments where, or drug discovery things where the genetics is as crystal clear as this, right? And there are very few of those that have been into drug discovery. And so, you know, we all talk about precision medicine. We all talk about the human genetics is finally going to make us have clinical decisions and drug discovery decisions. You're looking at one right now where you're taking, I'm being overly confrontational on purpose, right?
Mm-hmm.
We're taking shitty cell biology, overcoming the most beautiful human genetics, and we're using cell biology. And the only reason we listen to cell biology is because we used to be able to think about how to put a JAK inhibitor in a clinic. We need some cell model because we can't really be sure it's important for the disease, so we need to make some model. Here we have the opposite, where the genetics is crystal clear. And now you're worried about the shitty cell biology, not the clear. If you have two options, what do I believe? The genetics, right?
Yeah. I mean, I guess Paul Workman sort of spoke to that in that article where they're putting genomics or genetics as one of the highest things.
It's the beautiful experiment.
Yep.
I do agree with you, Al. Jungfu speaking here. Like that, there's a huge caveat that as that like the cell lines that have been established and culture, like come in the absence of the ligand. But the thing is, I do, in my opinion, it doesn't like hinder the progress if I concurrently I'm trying to tease out like more mechanistic detail, although it may not be 100% like physiological in a patient, in the context of patient, but like it doesn't harm to know more, right?
It's your life, man, but it's a waste of time characterizing weird-ass cell lines. I mean, it's an insignificant eddy in the river of science.
Okay, so may I just comment one quick? I mean, I agree what you were saying, Al. It's an interesting hypothesis or idea. But one of the other problems, Amit Nomai asked me a few minutes ago about the dose selection, right? Like, what is the dose selection with? I understand your, no confidence on the cell line and stuff like that. But what is the dose we are going to select?
The Fox model.
Okay, so from there.
So having in vivo efficacy is important.
Yes.
Some kind.
Well, just to get the dosing. I don't efficacy, no. Dosing, yes.
Yeah, that's what we have to figure out.
We need efficacy to determine.
Yeah. So we need some sort of indication. What is the dosing to go
Absolutely.
for the patient?
But you do have signaling. I mean, he's done a nice job here of showing that signaling, right, is correlated with target engagement. So that can give you a proxy for, right?
Okay.
Yeah, okay. Good discussion.
Can I just ask, this is Chris Jones from London. Can I just ask, what is the problem with these cells?
What did he say?
He's wondering what do you think the problem with these cell lines is the fact they're being radiated, I think.
I don't know. They've been selected for growth under this certain condition. I don't know what's wrong. Maybe there's nothing wrong with them, but it's, you know, the idea of relying on an immortal cell line that has
so they're not immortal. Just to be clear, they're not immortal. So some of the relatively low passage patient-derived cells grown under stem cell conditions. These are not classically engineered, immortalized monolayer cultures that you might be thinking about.
Got it. Okay, but they're still highly unusual, shall I say?
In what respect? They're not unusual in respect of DIPG patient tumor biology. They're actually quite reflective of that, at least at a very simplistic molecular level.
Well, to be fair, I mean, what we describe as, that's in the eyes, we're having fun here, right? But that's in the eyes of the beholder, right? One gets a cell line and then you look for features that correlate with something that you think one thinks should exist. And then you start to convince yourself it's the real thing. If these were reflective of true DIPG patients, I would argue that every single cell or every single stem cell from that patient should grow. And I've seen that the gymnastics that folks have to do to get these cell lines to grow.
That's just not true. There's no gymnastics required. These things actually tend to take pretty readily.
Well, Jerome was saying that it was, okay, that's not what, so in other words, from every DIPG patient, we could create cell lines like this, no problem.
Not every. I think in our experience, most people's experience, it's somewhere between 2/3 and 3/4, something like that.
And that's an interesting point. I don't know if you had heard what I brought up earlier. One of the things I kept asking myself, so a lot of these patients usually undergo radiation therapy. And then I'm wondering if the cell lines are derived from the patients prior to radiation therapy, where you actually know you irradiate, you probably kill the cells that are going to die, and then what's left behind, like a selection pressure. So would a lot of these cell lines be derived after radiation or before?
So the three you have on this slide were all derived after. They all came from autopsy specimens, but there are many, many biopsy-derived cultures available.
Hi, this is Angel from Barcelona. The Barcelona cell line was derived before.
Oh, well, sorry, Angel,
before radiotherapy. It was early. I mean, the patient died right after diagnosis, and this was an autopsy, but the patient didn't have time to get the radiation.
Okay, thanks.
So have you looked at, like, if you have a tumor prior to diagnosis and then you treat it with radiation, does the tumor change? I mean, have you done a lot of the evolution of the tumor to see whether or not it's actually different after radiation in terms of the genetic profile?
Yeah, absolutely. There are differences at diagnosis compared to autopsy. The kind of key driver mutations, and that does seem to include ACVR1, clonal and remain, but there are certain other mutations which are selected for and against for sure. Yeah, I mean, it's different after extensive radiotherapy and a whole variety of targeted therapies that are often thrown at these kids as well.
Okay.
We can provide both treatment naive and post-treatment models for this.
Yeah, and I think that kind of goes straight into some of the discussion, because it'd be nice, I mean, if we think like ACVR is actually one of those initiating mutations, and then with time...
That's another important point, actually, in the discussion about the genetics being pure and clear. It's pure and clear in as much as we do know the frequency of patients that have this mutation, so we can mark that subset of disease. We don't know what mutant ACVR1 is doing when the child presents with a DIPG. Some of the work of Jerome and Oren and others shows that it's definitely initiating, at least in combination with the histone mutation. We don't know if the cells are still dependent on signaling via mutant ACVR1 actually at the time you're going to be treating these kids.
Exactly, and so...
Has anyone had like knock-in mouse with this mutation?
Chris, can you guys comment on that? Like knock-in mouse models of...
This mutation, what it has in it? Like
Alto knock-in.
So, yeah, what was the question? Sorry.
Yeah. Has anybody done an ALK2 knock-in mutant?
Yes. So, Maria Castro in Michigan has a model, and there are potentially cells available then for xenografting and making a syngeneic model, which I think will come up in the later discussion as well.
Oh, what's the phenotype then? What do the mice develop after the mutation?
So this isn't my work, this is Maria's, it's unpublished, but it forms DIPG-like tumors if you have the right combination of mutations in there. And that's the other thing to remember, is that this is, I think, combinatorial genetics that we're talking about.
Yes.
This is not FOP.
Okay. Anyway, good discussion. I mean, we'll come back to this again closer to the end as we discuss again some of the strategies going forward. But it's very helpful discussion.
There's also another thing which could come up a previous time as well, which is the actual assays that you're doing here. So sort of how long are you running these assays for? Are they in 2D or 3D? And just a bit of refinement there might be worth discussing as well.
Okay.
The cells you got there have very different doubling times, for example. And that's a bit of some of these patient-derived models, the doubling time can vary a lot.
Okay. That's interesting.
Yeah.
Yeah. Hopefully we'll loop back to this discussion, because I'm also thinking about ALK2 as kind of a synthetic lethal. Like, I mean, it reminds me a lot of KRAS. KRAS initiate tumors in the background, but later on, other drivers take over. But then could now that same mutation kind of be a synthetic lethal? Back to some of the combination strategies later, by hitting that node along with other things. So we'll come back to some of those discussions. So you may not see anything with the compound by itself after a while in some of these tumor lines, but then in combination you see very, very robust effects. Okay, continue, Jean-Fu. Sorry about that.
No problem. That was an exciting discussion. So for my essay, I have optimized for the different DIPG lines so that like without the compound, so just with the DMSO control, so I perform like different titrations so that at the end of the incubation experiment, like without any compound, I'm not inducing like cell death or like stressing out the cells just by like having them overgrowing. So and only after that I use those conditions to perform the assays. So ideally, theoretically, so all these killing effects should be because of the presence of the compound and not just because they were overgrown after like so many, like more than 1 week of culture. So yeah, so maybe we can proceed to the next slide. So just because I've been like out of action for a few weeks, so mostly I'll be talking about what my plans are, and like some of them are ongoing as well. So in a previous cell assay, I saw that M4K 2163, although not the most potent ALK2 compound, but it has really nice killing efficacy, and on all of the mutant ALK2, all of the DIPG cells with the mutant ALK2, and also to some certain extent even on the one with the wild type ALK2, and will be interesting to also test out like the new compound that have just been shipped to us, M4K 2198, because the replacement of the middle methoxy group with the fluorine seems to have changed the property drastically. So yeah, like we also be looking forward to check whether the killing effect, whether it is just a general toxicity. So I'll be checking it in other unrelated cell lines and see whether how the effect is. Yeah. And definitely we'll be looking forward to know what the difference in the off-target kinase. Yeah. Yeah. Okay. And we can move on to the next slide. And among the DIPG cells with the mutated ALK2. The DIPG4, which is having G328V mutation, seems to be quite a bit less responsive to most of the compounds, and I think it would be interesting to know why. Listed in the paper are some of the other mutations they have, and so it will be interesting. I've gotten the plasmid for nanopore assays with the different ALK2 mutations from Omega. So it would be interesting to see whether it's something intrinsic. Although based on the biochemical data, they are equally inhibited by the compounds, but would still be interesting to see in the context of in the cellular context. And also I'll be checking also how does the downstream signaling pathway, how are they affected by comparing this particular DIPG cell lines with the other cell lines, and when inhibited by the compound. And yeah, I'll be getting the results soon, and it will be interesting to see.
Okay. Good.
And this will actually give clue to what kind of combinatorial treatment we should employ.
Yeah.
Yeah.
I disagree. I don't know. This kind of science is like, these cell lines have different genetic backgrounds, different signaling, grown in different ways. It's, I'm an enzymologist. In the old days, it was don't waste clean thoughts with dirty enzymes. And the idea of trying to disentangle signaling, interconnected signaling pathways on different cell lines with slight differences, with compounds that have off targets that you never know. I mean, it's not, as Medawar said, it's not that the question has to be interesting, it doesn't matter. And I honestly think that this is the rabbit hole that wastes time and effort, right? I understand we have a problem in the clean data on the one hand, the enzymology, the target engagement, the good chemistry, and the cell line data. And we could spend 6 months trying to sort it out, where we can do deep genome sequencing of all the cell lines. Then, you guys are going to be saying, let's do some RNA-seq to find out which gene expression profiles are different. And then we're in this little rabbit hole as kids keep dying, right? And so we have a molecule that's safe and effective and on target. We don't know what's going to happen in the clinic. We can never predict what combination therapies may or may not be effective. If we make that clinical tool available to our clinical community, they can start experimenting with it as well. And I think this is, I keep saying, the last I'll say it, I think this is a really big distraction for us.
Well,
I mean,
whether it's rate limiting or not. I mean,
yeah, I mean,
if it's done in parallel with, just pushing.
He's got a life, he could be doing other stuff, right? I don't think.
But either way, so even if we agree 209, let's rush that to the clinic, science is still going to happen in the background of ALK2, and so people are going to be learning stuff. I mean, so to your point, I mean, that science shouldn't stop. Even if we stop everything,
I'm just saying this kind of science is a dangerous science, particularly for a postdoc who's trying to ask a crisp question about biology. This will not, in my opinion, generate the kind of insightful answers. There will always be a question mark after this experiment. There will always be one. It's not the killer clean experiment. Clean thoughts, dirty enzymes. Think about that adage.
Okay.
Yeah. Thanks for the advice, Al. I'll keep track of my time, and
I'm thinking about you, dude.
Yeah, I'll know when to quit. Yeah.
Okay.
I wouldn't go down too far into the rabbit hole. Yeah. I'll at least try to grab it by the tail, but yeah, I wouldn't follow it too far. Yeah.
Okay, let's go. Keep going.
So we move on to the next slide. So all these are the way I designed the experiment is not that I'm going to like perform RNA-seq and try to catch like tiny details. So I've designed it in such a way that I'll be testing it like with once or twice Western blot, and if there is, then there is. If not, then I'll just like move on to something else. So like you need like yeah. So I definitely wouldn't go down the rabbit hole too far. Yeah.
Okay.
So
next slide.
yeah. So the next slide is that to also figure out why some compound are maybe back to the number 10.
One slide back.
One slide back. Sorry.
Yeah. So because compared to the pattern compound, our M4K 2000 series, most of them seems to be more efficacious, and I'm performing the experiment now to quickly check why, and this is by comparing some like good clue in terms of like the difference in the off-target kinase. So also like don't be concerned. Like this will be a fast experiment. I wouldn't like spend months on it. Yeah. Yeah. So can move on to the next one. And like Chris mentioned earlier, so all these patient lines, they are not homogeneous. So how's it? There are certain degree of heterogeneity. So for some, like for example, the DIPG 21. So with some compound, right? A population almost like half of the cells, they are really resistant. So this is a kind of I would think this as an opportunity to preemptively see what changes the cells could undergo to resist the killing effect. Like so I'm showing like just the feed. Yeah, it's not very clear from the picture, but yeah. And also this will be like just some Western blot and like some real-time PCR. Nothing crazy to just basically identify and characterize the population that are resistant. And then we may or may not then think of something to like overcome that. Yeah. And I think that's all I have. Thank you.
Okay. Thanks, Zhongfu.
No.
Thank you. All right. Okay. So to speed things up, we'll move on to some of this update from David. Which one is this one?
Yeah.
Okay.
Can I grab the mouse?
Yeah.
Okay, so just a quick update on where the chemistry is at. So again, this is the slide of the top compounds, which I've been showing you in the past, and I think Matthew did a nice job highlighting the fact that 2009 is our primary candidate, and 2117 and 2143 are the nice backups, some slightly different features. And we also have a number of other compounds in the pool that we could go back to, which have also been quite extensively characterized and could form backups to the backups, should we even have to go that far. 2146 was one of the ones which we thought would enter this pool in terms of meeting all the criteria up top, and something which we could take forward into some further profiling. And unfortunately, while it was a nice way to address the potential hERG liability by introducing the sulfonamide, which eliminates the hERG entirely, the PK was poor and the overall exposure was quite low. So this does not look like a good way to proceed in terms of mitigating hERG with the sulfonamide group, but we do have other things ongoing, which I'll come back to in that regard. This is a kind of a secondary set of the top compounds. These also show the same features that we've been after in terms of the biochemical and cell-based potency, and again across a number of different chemotypes. And 2163 is the compound that Zhongfu highlighted, and I'll kind of come back to that in a moment, also in some of the things that we'll be doing around that compound, because it does show some interesting results in a now much maligned cell assay. There's some compounds of interest that we're also calling this class of compounds which don't yet meet all of these criteria, but which do show some interesting activities and cell activities, and some in fact are not entirely profiled.
But we're viewing these compounds as something that, if with some further development, might go some way to, again, address the herd issue, and particularly 2184 and 2192. These are the compounds with the amide warhead, and this is a series that DIVA is currently working on to address some of the selectivity and PK issues that relate to these compounds, because they are very clean on herd, so that's a potential set to move forward with. Some other compounds which we are classifying as compounds of interest, and I think Sue is going to talk more about this in her presentation, are the indole containing compounds, benzimidazole containing compounds from Charles River. And here's a couple examples, again, showing some nice potency against ALK2, but some work in the nanobread and in the cell-based assay. So I think that's an evolving story still. So this is the data that John-Fu has shown before regarding 2163, and how it was actually quite efficacious in the viability assay across the board. And so what we want to do is we want to understand potentially, are we hitting some other targets with this compound? And so one of the ideas was to, of course, at the first pass, let's submit it to the kinase panel at RBC, 374 kinases. Let's see if something else is going on there in the background. So that's in progress. And of course, the idea then is to, and I'm sure John-Fu has this in plans, to try and repeat this compound to see if we can recapitulate this result in this set of lines. What's also interesting is that 3007, and again, John-Fu spoke to this, is that it's potent on ALK, as potent as any of the 2009 series, but we're not seeing any results in these lines. So again, one of the potential things we could do is look at 3007 in the kinase panel. And I think Alex and Stu have some interest in this also, just from the broader development perspective of 3007, just to try and understand what its kind of broader profile looks like. So that's something that's in the pipeline. In terms of the 2163 series, we now have a number of compounds, a number of analogs available, so they'll potentially be available for screening in John-Fu's assays. And we're also looking at making a couple analogs to kind of close out the series, a little bit of a different chemotype in the south, which we've known is efficacious in 2009. So get a little basket of compounds in this set, and see if anything can be done with them going forward. And that's pretty much it in terms of the compounds that are ongoing. As we see, we've kind of focused on the chemistry, and we're focusing on just a couple of the key series.
Yeah, that's good. Any questions for David? No? Go on to Sue. Sue's presentation, yeah. Okay, so you're up.
Okay, thank you. You go to the next slide then, please.
Yep.
Okay, so just a general update on where we're at. We've sent a further 5 new compounds through to RBC this month. The ones that we've completed this month are more analogues of the benzimidazole that Dave was just mentioning, and I'll talk about those in a moment. We've also been looking at some compounds that we had designed that were similar to the University of Houston patent. We've not quite completed the synthesis of these, but we've been making some progress on on those. Excuse me. But the majority of our effort has been focused on analogues around the blueprint compound to try and see if we can improve the properties of that compound to make it more, hopefully more brain penetrant. Completed synthesis of a couple of compounds that I'll mention, but unfortunately they weren't quite completed in time to go into the screen that was that should be running right now. So go to the next slide, please. Okay, so just talking about these benzimidazoles. So we had made, there's probably about 8 to 10 compounds that we had made previously. The highlight in this series was the 3045, which was about 21 nanomolar in ALK2, and was quite selective over ALK4, and so it's the one highlighted there in red. So incorporation of this for cyanogoat group did seem to give some significant improvements. And as David had mentioned, some of the changes and modifications to the R1 group on the molecule, which is methyl in 3045, shown some evidence that we might be able to get something better. So we've made these had only been made with R2 as H. So we've now made the compound where R2 is the nitrile to see whether that enhances the potency. We've also included a compound with a cyclopropyl methyl on there. Those two compounds are in the current RBC screen. In addition, we've just completed the synthesis of a couple of compounds where we've incorporated an additional methoxy group to see if that's going to pick up a further interaction and improve the potency of these further. These are initially with the R2 is H, just because of synthetic accessibility. If these show any interest at all, we can go back and make some more with the nitrile. Okay, so next slide, please. So I mentioned that right at the back end of last year, there was a patent from the University of Houston, which is shown on the left side of this as UH15_4, which was claimed to be pretty potent at ALK2, but obviously because these guys were looking primarily at RIPK2, obviously it still does have significant activity against RIPK2. So we had already previously started to design some compounds that were similar to, that appeared similar in structure to this. And you can see on this slide the sort of compounds that we're trying to make on the right-hand side. So the napthyridone is actually reversed. And you can see on the right-hand side, you can see at the top the dry docking in the absence of water. So the black compound is the UH15_4, and the pink compound is UH15_4, and the black compound is one of our designed compounds, which provides similar interactions. And in particular, when you go to the at the bottom, you'll see the water-mediated interaction between the carbonyl and the lysine. So we also believe that if you replace that benzyl by a 3-pyridyl methyl, that would provide has potential to provide improved selectivity over RIPK2, because RIPK2 has a leucine where this tyrosine is. We believe we would be able to pick up an interaction, water-mediated interaction to that. So next slide, please. So this is looking at the blueprint compound. So obviously, as we know, this compound, M4K 3007, which has been talked about quite a lot today, has pretty good, very good ALK2 potency and a good selectivity over ALK5. The downside is that it has quite poor brain penetration. It also seemed to have only moderate permeability and did show efflux in the CACO2 assay. So we believe that the high molecular weight of this compound and the high pKa are likely to be significant contributors to the poor CNS penetration. There's two potential basic centers highlighted here, which could be contributing to the high pKa. So we made a couple of compounds. The initial compounds were within the blueprint patent, which we discussed with Jeff previously, to see whether just modifying the structures slightly to reduce the pKa in one case and the molecular weight in the second case, to see what the effect of those were in vitro, and to see whether the we improve the permeability and reduce the efflux with these structures. And the synthesis of those has been completed, but as I mentioned already, they didn't get completed in time for the latest screening. On the right-hand side, in the first box, we've got a compound where we've replaced the piperazine at the top from the blueprint compound by piperidine, and that takes out that one potential basic scent, and synthesis of that is progressing well. We should hope to have that before too long. We're also looking to make some compounds where you've got a substituent on the far right-hand side where we've got the R group, and depending on the nature of that substituent, can influence the pKa of that piperazine nitrogen as well. Both of these areas are actually outside of the blueprint patent. If you go on to the next slide, please. So the other aspect that we've been looking at is how could we use the structural features of the blueprint but modify the core, and then we would obviously have to adapt which substituents we attach to that core to ensure that we got the pKa and molecular weight in the favoured region to give us a reasonable MPO score. So we've been ensuring that the targets that we make have an MPO score greater than 4.5. Synthesis of some of these cores has been initiated, and as I say, they'll be combined with the appropriate R1 and R2 groups. Okay, so if you move on to the next slide, Sylvia, are you on the line?
Sylvia? Oops.
Oh,
Sylvia?
Hang on. Okay, can you hear me now?
Yes. Yeah.
Sorry. So thanks, Sue. I was just saying that this is a comparison between the structure that has been previously described by SGC in Oxford and what was made available in a conference from the Blueprint Medicine. We have the R2 bound to blue 782 on the left side and our crystal structure on the right side. What I want to point out is that effectively the two ligands are binding in the same way, and the water network that the SGC was able to solve is effectively identical to the one reported by the Blueprint Medicine. So it's definitely something very conserved across these kind of molecules. If we can move to the next slide. What I've then done is calculating the stability of these water molecules, and I use a program called the Water Map. What effectively does is calculating how, let's say, stable or happy these waters are in the binding pocket, and then it colors these, it color-coded these results from red being red, very unstable molecules, or green, very stable molecules. So here you can see that we have two kinds of data in the middle panel. We have data from the results with from the calculations without the ligand in the binding pocket or apoprotein, and pyramids points that are results with the ligand in the binding pocket or holoprotein. So what we see is that these waters are all embedded in a rather extended hydrogen bond network. But there is one water, the one that is sort of the first one closer to the gatekeeper, the threonine gatekeeper, that is particularly unstable, apparently, even though being engaged in bonds. This is, of course, something, is a calculation, so we are not, I mean, there is always a little bit of uncertainty, but in terms of potential opportunities to functionalize an inhibitor, these could be a water that can be sort of targeted with hydrogen bond acceptor and donor groups from the piperazine ring of the blueprint scaffold.
Okay, that's good. Interesting.
We believe that, we think at least that these non-stability is due by the fact that effectively the piperazine, which is close by, the piperazine from the blueprint scaffold is presenting the adipophilic part to this water, and therefore is the water itself is not very happy. And you can see from the distance with carbon, we have roughly 3.8 angstrom, so it's rather close. What we are currently doing is investigating whether this sort of water network or water in immediate proximity to the threonine is also found in other kinases that have a threonine as a gatekeeper. And then we'll also look at potential substituents around this area.
Okay, it's good. Okay, that's it. Any other questions for Sue and Sylvie? Okay, thank you.
Thank you.
Okay.
Thanks.
Okay, so I think we're right on time. So what I'm going to do is go back into some of those, exciting discussions we had earlier about more strategy, and then I'm going to throw some provocative thoughts out there for people to kind of chew on a little bit. So what I thought I'll do first is, so everybody's aware that I think on Tuesday a paper was published recently in Nature Communications, and that sort of kind of allude to some of the things that we have been discussing. So this paper by this Betcher's group in Chicago, I think. And so what I've done on the next two slides is more or less summarize some of his findings, because this lends nicely into some of the concept behind combinations that we've been thinking. So the paper was in Nature Communications. This was the title of the paper. It got published on, when was it? On March 4th. Okay, and they're kind of emphasizing the fact that the ALK2 mutation, particularly R206H, cooperates with the histone mutations to promoting the pathogenesis of the disease. And so what I thought was interesting is, you could say on the first panel here, B, that, the ALK2 mutation alone, does kind of affect the survival of the animals, but some of the other lesions don't do much. But when you combine the ALK2 mutation with the histone mutation, start seeing a spread in the survivability. So, I mean, there's no question that the pathogenesis of the disease really rely on the combination of some of these lesions. So I just jot down some of the key take-home points here from the paper. I mean, so they're saying that the ALK2 mutation to R206H accelerates DIPG without the histone mutation, where the histone mutation actually requires the ALK2 mutation in the background to drive the disease. So that's kind of an interesting thought process. They were also able to show that inhibition of ALK2 alone with some of the known tool compounds that are out there were not sufficient or robust enough for the anti-tumor effect, again suggesting that combination might be necessary, to treat the disease. And so what I did on the next slide was I highlight some of the compounds that they tested in some of the cell lines. And what they found is A204 is one of the compounds that they tested. This compound had activity in some of the mutant cell lines, but because the other compound, which is also selective at ALK2, didn't, they were wondering whether or not the activity that they saw with 8504 was coming from some other mechanism. And so more and more, I think Jean-Fou related to this, but we tend to refer to compounds that are active that we're not convinced is ALK2 as off target. But I'm starting to think whether or not these compounds are teaching us how to actually kill ALK2 cell lines. So maybe in addition to ALK2, if we understand what that other target is, instead of calling it an off target, it actually might be a target that might be necessary to actually be an Achilles heel to some of these tumor lines. And so, like Aled was talking about, using 209, which is very selective, understand what that is. But maybe we can let these compound teach us actually how to kill some of these tumor lines. So keep that in mind. We might want to understand what other targets is 854 hitting that might be an Achilles heel in addition to its ALK2 activity that might be killing these cell lines.
Just to say that in our hands, we do see some potency and selectivity for 214117 in ACVR1 mutant DIPG cells.
Okay.
What level of selectivity?
Varies depending on the line you use, but 2 to 5 fold compared to wild type, something like that.
Okay. Okay. That's good. So the next slide, what I try to do here, in light of the view that we're thinking about combination. Hello? Sorry. The fact that we're thinking about combination strategy, in addition to the ALT mutation, so we could use a selective compound. I'm thinking that one of these strategies is to try some two-dimensional plots looking at combination. And then we can look at EZH2 inhibitors to see how those synergize, just basically knowing what the histone mutation does. Looking at some ALT2 compounds in combination with EZH2 inhibitors, and I've just picked the GSK compound and the Epizyme compound as examples here. The caveat is that those compounds are not CNS penetrant, so that would be a challenge. However, compounds that work along similar mechanisms are the EAD compounds. You know, those are more allosteric to the enzyme binding site in the PRC2 complex. Those compounds are more CNS penetrant. So if you see evidence of efficacy with an EZH2 combination, and then more so we see it with the EAD compounds, and the EAD compounds afford us a path forward in terms of having CNS penetrant compounds that might actually work. Javad and his team have also shown that panobinostat actually works very well in killing a lot of these cell lines. However, that compound is not CNS penetrant, so right now in the clinic it's being utilized by a mode called convection-enhanced therapy. However, there's a compound that's out there that's an HDAC inhibitor called compound 26 that's very CNS penetrant. We have it in-house. It's not commercial. We have it in-house. So we're actually going to also test it in combination to see if it does work. If it does, again, we have an example of another CNS penetrant HDAC inhibitor that might be able to combine with our CNS penetrant ALK2. So that's looking at combination of epigenetics. So we're planning to start out with, pick two of the cell lines. I mean, Jerome, we got guidance from Jerome in terms of 007 with the R206H mutation. And then Jerome has this other cell line called the DIPG36. We thought those are two interesting cell lines to start with because he said those ones in graph okay in mouse. So it'd be nice that if these actually work in terms of the synergistic study, we actually can rely on the possibility that we might be able to do a mouse model with them. In addition to the epigenetic targets under consideration, there's a lot of other kinase lesions that tend to get, you know, deregulated in a lot of these DIPG patients. And we're talking about the PDGFR, you know, amplification that's accompanying a lot of these tumor types. And quinolinib is a compound that's out there. But more recently, Blueprint has a PDGFR inhibitor in the clinic. It's actually not bad. So we have those two in-house. We're going to try to see if those combine well with ALK2.
Just to say that in DIPG patients, you very rarely see the PDGFRA amplifications or mutations in the ACVR1 mutant subtype.
Oh, okay.
Almost never. It's almost always in the H3.3 K27M subtype.
Mm-hmm.
And the same with the CDK4 and 6 amplifications. They're very rarely seen with ACVR1.
Okay. Interesting. So it would be interesting to see if ALK2 inhibition could serve as an Achilles heel to some of these. So we'll just run the combination and see if we see any hint of synergism. Yeah, if that's reasonable. And then you got some of these cell lines have PI3 kinase mutations in the background along with mTOR. So GDC084 is right now a compound in the clinic that's CNS penetrant. It's a CNS penetrant PI3 kinase mTOR inhibitor. It's looking good. So we'll try a combination of that with our ALK2. And then the old-fashioned everolimus is being studied also. And then more recently, I'm starting to know, since if ALK2 kind of look at deregulation along the BMP axis for signaling, there's always a bigger question about the TGF-beta signaling axis. They're all playing a role in driving tumor growth. So I'm thinking of adding to this mix, you know, the TGF-beta signaling, see if these would create an Achilles heel to these tumor types. So, and this compound is in the clinic currently. So that's another kinase inhibitor we want to test combinations with. Okay, so those are some of the combinations that's planned now. Jerome was going to kind of speak to that in terms of the experimental details, but he couldn't make it today. So next meeting, he'll actually map out the plan as to what he's actually executing. And so you'll see that in a little bit more detail. Okay. Now, the next thing I wanted to move on to is, again, it's a thought process that came up recently. Oops. And so, oops, how do I go back out? How do I go back out? Yeah. Okay, so we've all talked about, alter mutations being essential to driving some of these DIPG tumors. We know that it occur in combination with this histone mutation, and then there are all these other lesions that can accompany the disease. But more recently, I realized that histone mutation apparently have been shown to produce antigens. Antigen-specific T cells have been isolated from some of these patients, and that has triggered a sort of clinical study looking at a little peptide fragment as a vaccine in some of these patients, which now have us considering the immunotherapeutic approach to probably looking at some of these DIPG type of tumors. So with this in mind, I actually went and started looking at all of the clinical trials that were ongoing in DIPG. And it was interesting to note that some of the PD-1 inhibitors that are out there, it's creating a lot of traction in immunology. So the first one was just recently approved by Sanofi Regeneron, this REGN2810. It's currently in clinical trials, and I actually highlight the clinical trials numbers here. It's a phase 1, phase 2. It's a PD-1 inhibitor, and they're treating patients right after first-line therapy, which is radiation, to see what these type of inhibitors would do in some of these patients. Similarly, again, Bristol Myers Squibb is also looking at Opdivo. Hi.
Hi, it's David Jenkinson here. I think that this is a dangerous route to take. There was some. It's possible that the checkpoint inhibitors are useful post radiotherapy when you've debulked the tumor, but there was some data shown at ISPNO last year from Eugene Wang, and he was showing that actually very small numbers, 5 patients treated, none of them made it past the second dose of pembrolizumab, and they all did worse than historic controls. So his view was that it's possibly due to the location of the tumor. You've got rapidly growing tumors in quite a restrained place, and then you're getting an influx of immune cells, and that was causing rapid progression. So you may not want to do immunotherapy, except in possibly in the post-radio window where you see, where you've debulked the tumor a bit.
Okay, that's interesting.
So, from my perspective, those tumors are usually not debulkable, and the majority just undergoes a biopsy, but I totally share your concern and the
So
problem.
radiotherapy debulking rather than surgical.
So
the
area where those tumors sit is so tight to begin with, and that every additional inflammation is putting that child's life at risk with a post-radiation inflammation that takes some time to wean off. And in this regard, I absolutely share your concerns. We have currently an open trial with Ipi/Nivo 6 weeks after the end of radiation, and there are concerns as well from it, and it has caused major challenges for these children due to exactly those concerns that you raised. And as well, there are opinions that really these are not T-cell triggered tumors, and that in fact from a really scientific perspective, it entails danger and less really hope that the mechanism is valuable.
Interesting. That's an interesting field, because the way I ended up framing the question, so based on this therapy that was in the clinic, I started kind of just framing another question. So right now for the whole program, we were thinking of designing selective ALK2 inhibitors, okay, over ALK5. But then the more and more I learn about the TGF-beta signaling pathway and the role it plays in immunology, whether or not we were eliminating candidates that might have potential immunotherapy. So that's how the thought process. I kind of coupled to the PD-1 therapy that was out there in the clinic, and then looking at our strategy. And then the question, what are the risks versus benefits of targeting ALK5? That's the way I framed the question. And so this was just some of the background that was out there. So right now, I mean, Lilly has a compound in the clinic, an ALK5 inhibitor. So there's proof of principle that TGF-beta inhibitors actually are anti-cancer types of targets. So the compound is called gallonesertib. It's orally administered, and it's right now given in combination with checkpoint inhibitors. One of the things that's known is that TGF-beta strongly inhibits T-cell proliferation and negatively impacts the function of CD8 and NK cells, and they do quite the opposite to the Tregs and MSTs, exactly what you'd want in immunotherapy. So that kind of triggered the thought, maybe ALK5 inhibition that we know is important as an anti-tumor strategy. So then I started to ask myself the question, and I mean, some of it is actually shown here, some experiments. You could see that if you take this is some studies done by Lilly. If you take CD8 T cells and you actually expose them to TGF-beta, you could see it diminishes, it suppresses the proliferation of the CD8 T cells. But if you now add the TGF-beta inhibitor, you can see that proliferative profile of the T cells come back. The same thing happens if you take naive T cells and expose them to Tregs, which actually produce TGF-beta. That TGF-beta produced from Tregs, actually, you could see they suppress the naive T cells, but if you treat them with the inhibitor, you recoup that suppression of proliferation. So that was exciting from an immunotherapy standpoint. So what I did then, I went and I started looking at some of the preclinical models, and it was interesting to learn. So this is Lilly again. What they had done is taken the ALK5 inhibitor, and they show that, you know, if you combine this ALK5 inhibitor with a PD-L1, and this was done in a syngeneic mouse, CD26, okay, it's a different tumor type, where you could see, I mean, the two compounds by themselves, here in this quiz is the ALK inhibitor by itself, here is the PD-L1, but when you combine them, you actually flatten out the tumor. Similarly, BMS have an ALK5 inhibitor recently. They show, so in green is the PD-1 therapy alone in this MC38 syngeneic mouse. Up here, the compound by itself, but when you combine them, you could see you flatten out the tumor. So you got two distinct tumor types hitting the same target and actually have the same outcome when they combine them with immunotherapy. And it was interesting to learn from some of the readings, and some report from Lilly too. They found that a lot of the patients who don't respond to a lot of the checkpoint inhibitor therapy, they found that the tumors have a lot of TGF-beta in them. And so it appears that TGF-beta actually contributes to what they call this immune-excluded phenotype, okay, which leads to, okay, inactivation. So however, when they combine these tumors with TGF-beta, ALK5 antagonists, you know, the tumors now become susceptible to the immunostrategy. So with that background, I went and I started looking, okay, what sort of ALK5 inhibitors are in the clinic? And so gallonesertib is in phase 2. Sorry. So this compound from Lilly is in phase 2. They have a backup candidate also in phase, so that's looking very good too. And then you have these other compounds, one from Pfizer and this from a company called Medcactor. So they're all in phase 2. Now, one of the concerns that people had with ALK5, and that's one of the reasons we were trying to avoid it, in some of the early studies, ALK5, you know, appeared to be creating these cardiac lesions of valvulopathy in the beginning. And so people were very, very concerned. But they realized that that's actually dose-related, and most of it used to happen at the high dose. So, and it's something Lilly actually studied quite a bit. So they had to go back and find out if there is a therapeutic window that can be achieved. And they did accomplish that. That's why they end up going back into the clinic, you know, defining a decent dose window. So with that in mind, it's kind of like people are learning how to dose ALK5 inhibitors in the clinic, you know, diminishing the cardiac liability that usually exists.
Phase 2 trials in adults.
Yes.
These are all adults. Right?
Yes, these are all adults, yeah. Okay, so with those background in mind then, I mean, this was kind of the hypothesis with regards to ALK5 and ALK2 combined. I mean, this is actually animated. So what I had in mind is basically DIPG has, you know, ALK2 lesions, they have mutations. And then I'm wondering if, in addition to those ALK2 mutations, the fact that there are other lesions in the tumor, could we actually come up those other lesions to an immunotherapeutic strategy? So the idea is if we can, you know, combine an ALK2 inhibitor and an ALK5 inhibitor, then, you know, we might be able to actually get the required effect on the immune cells at the same time blocking the DIPG lesion. And so an ALK2 inhibitor should stop any ALK-driven DIPG tumor cells, and hopefully the ALK5 inhibition should block any TGF-beta mediated resistance if we had the immunotherapy to it. So we're kind of trying out as a proposal whether combining ALK2, you know, ALK5 inhibitors, compounds that have the dual activity with a PD-1 blockade, whether these would demonstrate anti-tumor properties in cells. And so if you look at our collection, because we were driven by identifying selective ALK2 inhibitors, we don't have very many dual active compounds in our collection. Well, like David mentioned, this is one of the compounds I found in our collection, 2192, it's 5 nanomolar at ALK2, it's 46 nanomolar at ALK5, which is not a bad profile. So these might be worth actually testing in some of the models in combination to see if we can actually synergize with PD-1 therapy. So that was kind of some of the thought process, but it's interesting to hear some of the clinician perspective as to the risk, you know, presented with using immunotherapy in the clinic. So those more tend to be thought-provoking, and I really welcome some of the feedback some of you just provided. So I think that was it. So in the end, we were thinking of just expanding a little bit on this of that dual-acting compound, looking at combinations of this dual-acting compound in some of those CD8 proliferating cells. And then I don't know if there's any syngeneic DIPG model that people have developed so that we can actually start looking at some of these immunotherapy strategy preclinically. So those are just some thoughts to throw out there, and also many feedback people might have on some of those.
So this is Al again. This is another rabbit hole, I think. We have a very, again, start from the beginning, we have very clean genetics. I would think that our role as M4K is to make a safe, on-target molecule for ALK2, and not to do all of this ourselves like we're a little Pfizer, but to allow that compound to the clinicians that they can test their hypotheses, which are going to be much broader than we can ever think of. But if we do our duty and provide the molecule, the terms that the clinicians can do anything they want with it, provided they get past their ethical and safety boards, to test it as a monotherapy or to their intuition and to their genius to figure out how to do, any combinations. Obviously, the EED is something that's obvious to check. But if we, as a small little group with a few clinicians, start to think about every possible experiment and going sideways to test this and that, it will distract us from getting a safe molecule that we can make available as a clinical compound, to collaborators all around the world to enable them to test hypotheses. We have lots of really smart folks from the UK on the phone. Imagine, if they had the ability, without having to sign their life away to AstraZeneca, I'm making AstraZeneca up, right? To do investigator-driven trial of whatever they wanted to do. I think that's how we could most quickly have a big impact on the field, as opposed, these are great ideas, of course, Method, but can you think about, how we can in the whole community of scholars around the world, many of whom are on the phone, how can we most rapidly get an impact to enable them and all the brains around the table? My gut is telling me the fastest we get to a monotherapy, safe, effective ALK2 inhibitor, the more then we enable folks. It's just the way, I don't know, I'm curious. That's strategic, but yeah.
David, I think my general view is I would generally agree with you. Maybe I should caveat that with this on this point. But I do think we've got to be careful that we don't bring an inhibitor to the clinic that, you know, we all know the history with kinase inhibitors, that there is an ability to for resistance to develop, or be it intrinsic or acquired, and we therefore get a clinical trial that doesn't show efficacy because we haven't done some thoughts around combinations. So I would totally agree that, you know, we are a small group that really has a focus on delivering, in this case, an ALK2 inhibitor into the clinic, but we've got to make sure, we've got to believe that if we do that, it is going to have a signal.
How are we going to know without testing?
Well, I think that becomes the next question. What is it that, talking to the clinical group around the table and starting to talk that, what is it that we can provide? I mean, we're going out to do some checkerboard or synergy studies, et cetera, in direction from that. But what is it that would make you consider this as a combination, or reach to this, or give you confidence that you would get a signal actually in the clinic? What can we do as the next step, either within the context of, I'll be completely blunt, we have a grant that says we will do X. And, you know, we had everyone on the phone. I could probably write the final report saying we've achieved X right now. Now we have a couple compounds which I think are fairly potent, fairly selective, show brain penetration, show something that could be, you know, PK. We've met that. We have some time and we have a little bit of money. Not a lot, but a little bit. What can we do in now that drives this program to be a little bit more of a therapeutic, that gives us a clue of what we should apply for next? What's the next grant or next program we're going to all get around and say, we've done this on its own as a single agent, and we've done it in combination, we've seen these signals. We need money to take it to some step that a clinician is going to say, hey, they're onto something. If they get good data from those experiments, we can move forward. And that's kind of my question. I don't disagree with anything anyone's saying here. I'm just trying to say, I'm throwing my finance hat. I'm going to stand up with that, hopefully, in a few months and say, hey, this is our next, this is where it goes next. This is worth funding because we see something and this is the right way to go. And I'm trying to figure out what that is.
I mean, what we could do is help. So even though we know all of these cell lines probably may not be ideal in what we want them to be, in terms of, that's why, you know, I went to the clinic and find what compounds are out there in DIPG in the clinic that we can combine with, because it's going to be easier for them to transition, taking an ALK2 inhibitor that we might want to develop. So if we generate any sort of preclinical data that guides them to, hey, here's a potential combination that might, I mean, that's enough for them to say, hey, you know, these cell lines aren't the best, but at least, you know, they derive from DIPG and we're actually killing them. So this might be a viable combination that might work. I mean, it's just another way in terms of which combination strategy with your ALK2 inhibitor would actually be the best one. I mean, you know, not to hold back. I mean, 009, the single compounds still have to be developed on its own. We got to make the decision. One, we got to do rat tox, we got to do dog tox. That's where the costs start building up. So we got to decide, okay, do we push all our money, just get that going? In the meantime, people are going to use the tool compound to develop combination studies. It's not going to stop 009. If we believe 009 is a candidate for clinical development, that shouldn't stop us from actually going down a development path. I mean, it now starts to involve a lot more costs. Now the question, Owen has to do is,
are they going to do that?
Yeah.
put it in. They put HDAC inhibitors in.
Yeah.
Those are poisons.
Different aspects. You're right, right? So one aspect I agree with you, Alice, that you would want to make sure that it's a therapy that is safe. However, we look back of 30 years of trials with monotherapies that all failed, and these tumors are heterogeneous tumors, and the likelihood that maybe an active compound is going to be dismissed is sadly high, because we know that ALK2 is maximum. There are different data. Our data at 6 kids showed maybe 15%. Chris Jones' data showed 25 to 30%. So let's say at a good risk, a third may respond, but that will likely not show in a clinical trial traumatic prolongation of life in order to proceed forward. And I do think that the clinical community is open to and is convinced that only compounds will make a difference. This is as tragic as the disease is, but because it's so fatal as well, I think the likelihood to be open to do a study that is at least safe in so far monotherapies and maybe add additional components, I think is high. So I think as a clinician, I would absolutely advocate for a combined medication. How should this medication look like? So immunotherapy is making those tumors swell in a tight area, highly dangerous, and needing huge doses of steroids per se as a disease and in that context. And steroids are counterproductive to immunotherapy, so I would not go the direction of immunotherapy. I would look back to the biology of those tumors, and there is, aurora kinase B that was shown to be in a not insignificant amount of people expressed, and alisertib is an aurora kinase inhibitor, rather like more directed towards A than B, but has some B component. I'm not an expert in this field to express how likely there is a purely aurora kinase inhibitor B, but this is, to the best of my knowledge, only so far in adult trials. Alisertib has been used in a phase 2 trial with neuroblastoma, and as such we have a dose established as well as a safety profile, and as well in combination with other therapies. Temozolomide, you have that, right? Temozolomide and irinotecan. So I do think that a combination to prove preclinically a aurora kinase inhibitor with ALK inhibitor would be a promising combination.
And a preclinical model would be, it's not right, cell lines, mouse model, what I know Al's shaking his head every time I say anything because of the word cell, but it may be the best we have. I mean, it is what it is, and with all its, but it's what we have.
But like I was also saying too, so we have our selective ALK2 compounds that we set out to do, and we deliver those compounds nicely. One of the things we did too at the same time, again, back to the cell lines that Al had ate, these are derived from patients. We had some compounds that hit ALK2 that were killing them. So instead of ruling them out, we actually now have some compounds. So we can start thinking in terms of endpoint therapy. Some of these compounds have ALK2, but they probably have other lesions that they hit that might be beneficial to the disease state. So it's something to be open-minded. I mean, we have our ALK2 sound strategy. We have our compounds. I mean, put it in some of these cell lines, which are no good. It didn't work. But then at the same time, people might get enticed by, hey, we have some ALK2 inhibitors that are actually also killing the DIPG cell lines. And so David mentioned a couple of them. I mean, like 2163. We shouldn't actually rule those out if you're looking for endpoint efficacy. I mean, so those are some of the things to keep in mind as we go forward. I mean, So we got 009, and then we have some actual follow-on from that. So it actually looked decent in the cell.
But I want to stress that I imagine this experiment we're doing, M4K, as a experimental medicine project, not a throw poisons at kids and hope it works project. If we put dirty compounds, dirty meaning non-selective compounds like HDACs into children, and it doesn't work, we are none the wiser, we've wasted money, etc. By making as clean a compound as we can to a mechanism we know is active, and enabling the community to test hypotheses with a surgical strike as best we can compound. Even if it fails and we get resistance, we'll know why, and we'll have a strategy, much like BRAF went that way, much like all the other kinase monotherapies. When you understand resistance, then in the clinical side, you can design new molecules. And so it's, I believe, a riskier approach to science, remember? Because we keep bemoaning the fact that 30 years of oncology, it doesn't work, we don't know why things are working. It's because decisions are made based on, you know, non-crisp chemistry and biology. Here we have the opportunity that the genetics is telling us a dominant mutation in kinase. To enable the field, if we make a highly selective inhibitor of that very same kinase where human genetics is telling us it is evolved somehow, maybe in initiation of the tumor, so maybe it doesn't work when it's a tumor, but we've got to test that hypothesis and provide the clinicians the tools to do experimental medicine, not drug discovery. So it's subtle, but
actually,
I mean,
one second, if I can just throw one. Al, I did agree with you very much so, and actually, I think we are going to be directed by what to do on this, just by also time and money. I agree, and it is of interest that all of a sudden we have a compound that we didn't really think was going to be that active, and this seems to be killing things. That may be an experiment for someone else to do. But I think there's a couple things we can just do. Find out if it's just toxic. I mean, we may be pouring bleach on this thing for all we know. But just, you know, find out a basic thing, put it out there and say, by the way, someone should look at this. How can we help? This is what we've learned. Put it out there. You know, we're going to run out of money and time to do that. We're focused on this. Agreed. We can only come up with, I think, if we're going to do, especially if we're going to do looking at combinations, we have to have something that's very clean so we can precisely say it's not something else that's happening. That's just,
I mean,
finance guys, believe it or not.
So the question, I mean, the thing I got concerned, so we have 009, which is a good tool. Now, if you look at a lot of compounds that gets approved going after certain targets, at the end of the day, it's safety and efficacy, which is the ultimate driver. I mean, a lot of very, very selective compounds they just don't make it. They're just not efficacious enough. You're going to learn about the target.
But they're never genetically dominantly mutated. That's the big difference. This is different. But
we're hitting that mutation. So that's why I say, if you get a compound that hits the mutation, it might all, I mean, you know, the old story, back to Clevic and all those guys. I mean, they hit the mutation that we want to hit, but they don't hit other things that contribute to the overall efficacy. What I'm saying, we have 009, it's very selective and we could learn from it. But in terms of a compound that's going to give you efficacy, you might want to have ALK2 along with something else that's safe. I mean, and we don't know, we may have the compound. I mean, 2163 and 009, the difference between them is minuscule. I mean, we might actually do the profiling and realize they're just as selective. Yet, we don't know. One of them has good efficacy in its own way and the other one doesn't. So, I mean, I'm not against going forward with the selective compound, but we got to be aware, like, it's going to cost. And if we feel like, okay, let's shut everything down, put 009, I mean, you got to run the risk. When you're going to development, things happen. So if you want to have fallback candidates.
Sure. Well, I think we're going to have, we have the time and money for fallback. I think we have a couple that are quite interesting, to be blunt. Yeah, I think there's one or two that might be more interesting than 009 personally, which I'd like to hear the thoughts later on. But having said that, I think, you know, I wouldn't mind hearing what the people on the phone have to say about what follow on Ute's comments about how do we position this program with 8 months left to go, so that it is well positioned that we can pass on good science to other groups to follow on, be it, as you said, we have a compound that kills a lot of cell lines, which, you know, that's a good thing, but it may or may not be as selective. But how do we position it so that we can go forward and produce a program that's interesting to clinicians, that they are saying, hey, they're onto something, let's raise some more money around this and push it towards the clinic. And so that's where I'm coming from on this is take a couple of our selective compounds, take some of our money that we have, do the combination therapies, and see what comes out of these things, see if we improve it. I think, and Al, please don't throw anything at me, but we have these cell lines, it's all we have. If someone has any other better idea for something that can give us an indication, I think we're really excited to hear it, but it is what it's the tool we have. Am I missing something? And we go from there. The gang from the UK, have I summarized kind of where we are?
Yeah. Sure, Ryan. I think it's two things. How do you plan to distribute the 2009? You know, you mentioned the 42 grams you would distribute, and then maybe you need to talk to clinicians a bit more widely.
Yeah, we'd like to do two things. So the short answer is, and Methvin, congratulations again. If anyone needs the compound, please contact Methvin for now. Arij has contacted me, and we're trying to put up a page on our website to give more information about the compound and how to use it and some of the characteristics and trying to charge people. And we would like to encourage it by word of mouth as well, and social media, etc.
It's an oral compound.
Yes.
It's an oral compound.
Yes. Yeah, that is part of our target market, our target product profile.
Yeah. I think that's really important aspect.
Yes.
And, you know, again, from a clinician perspective and from what we learned about those biology, is there anything out that would really, because the RAF pathway is certainly at the core of those tumors as well.
The RAF?
The RAF. RAF. So if there would be anything, I know Al doesn't want to hear it, but
But what's the evidence when people say pathways are involved? Because as a molecular biologist, it's quite easy to show that manipulating pathway X, Y, or Z will affect some cell.
Yeah.
Right? So I'm looking at the situation much like in most of the drug discovery world now, where human genetics gets 10 stars and all other data get half a star on the terms of how much you influence.
Yeah.
And the beauty of why we actually chose this disease is that the genetics is fairly clean, right? And I'd like to keep focusing on that because that's unbiased. That's not influenced by poor experimental design by some scientists around the world. That's not subject to confirmation bias or poor reagents. It is absolutely right. Then if you look in historical and drug discovery, like melanoma and BRAF, molecules that molecules that target genetic mutation do work. You get resistance, as David was saying. That's something that historically we've dealt with post facto. Put in the clinic, find out the resistant design molecules, and to David's point, maybe we can compress that, realizing that as the antibiotics guys, it'll happen. So this is why I'm cautious about expanding to other pathways and other things, because there's so much literature out there. We can find evidence of this pathway, this pathway, and this pathway. And maybe it's right, but we can't test it all. So the art of the possible is to make a very clean compound and share it with your colleagues who want to test RAS, and share it with the colleagues who want to test Aurora. That's why I'm so
passionate about it.
Yeah, because.
But I'm not aware, with respect to really MEK or BRAF inhibitor, how much it was not anticipated, this incredible response in low-grade glioma, right? This was not anticipated based on the models. It was, and those are pathway medications that are pathway driven. So how much of it is really something that is giving us certainty that what we find preclinically is really translatable? And
so interesting, you just said the MEK that is successful in the clinic was not efficacious in the models?
No, my question was, how efficacious was it? How much did it really translate? Because it's my impression that the success story that we saw, fortunately, in the clinical day-to-day success was not anticipated with the preclinical testing. And it is absolutely the exception to the rule. When you look, you just really mentioned the examples that one drug makes a difference in oncology, right? We would wish that Clevex, that sirolimus, that BRAF inhibition could be translated, one serves it all, but that's the exception to the rule. But I hear you, and from a clinician perspective, I think I may just really approach that topic from, it's very different when you introduce a medication that is potentially harming in a population where it's about improving quality of life, where it's about a survival chance where you destroy it. We have a low threshold in children that come after, 6 weeks of a bit of squinting and a bit of balance issue to our practice, and I need to disclose that they will likely be dead in a year's time. That's a tragedy that is just really so terrifying that you would want to not wait so long and making those, duplicating these efforts over the last 30 years where there was, the one drug, some evidence as well, but none of it really was fruitful. And
I only caution that this instance is driven by genetics. Most of the oncology stories in the past were driven by cell studies on pathway inhibitors that are up or down or linked. And that quality of evidence, as you say, or the burden of proof was much less in those other examples. This disease was selected because the probability of success of, I would argue, 2 therapies, one that hits the epigenetic arm and one the kinase arm, is, that's what the data, it should work. And to do that, you need to get safe and effective molecules for each branch, and ideally have some preclinical data, obviously, ideally. But in the absence of preclinical data, we shouldn't spend our time trying to squeeze to find a preclinical data that we can test it. Whereas I would argue the evidence is good enough to take these molecules into, if it was my child, I would look at the data and say, you have to test that hypothesis, right? So anyway, this is the cool argument we have to have, right? Because we're not going to go to a regulator and say.
No, I think we're at the right stage where we have data about a series of different molecules. We have, we actually have molecules to talk about, which is what we trying to have.
We have about 1 minute left, so.
I need to make one quick announcement. Eileen Shore, who's running the FOP model down at the University of Pennsylvania, she's not available. We're scheduled our next meeting for April 10th, which is a Wednesday. She's not available that day, and it's probably the most interesting data we're going to have coming out for the next while. I'm proposing that she is available on Friday, April the 19th. I know it's a bit of a delay, but I'm going to put out there that we move to 10 a.m. on Friday, April the 19th. Is there anyone on the line who is violently objecting to that?
The Brits are a religious nation, and it's Good Friday.
Is it really?
The Canadians are more secular. We don't care.
It's Good Friday, April 19th.
According to my library, which doesn't lie.
Yeah, you're right.
Yeah, okay, I didn't realize that. That might.
It depends. We'll ask the Brits on the phone. I won.
The alternate date is April 24th. That's getting awfully late in the between meetings.
But she'll be able to share the results. email.
She could just make some slides and, hey, I could present them and you'll get nothing out of it, but I'd love it if she was available to talk about the model, talk about what she saw, talk about next steps, talk about the weaknesses, strengths, et cetera. I mean, it's always better to have the person run the model there. So, let me put it this way, I'll throw it to the gangs at Charles River and the University of Oxford to pick whether the 19th or the 24th is better for you.
19th is a holiday here.
19th
Yeah, it is a holiday. Good Friday is a holiday.
Yes.
Despite the secular Canadian.
Okay, then the date that we have is really April 24th. I'm not gonna drag everything in on all.
I can't make the 24th, but don't let that stop you.
I'm sorry?
He said, don't worry about me.
I can't make the 24th, don't let me stop you.
I do worry about you, Alex, though. Okay, that's a long time between meetings, but it again is, I think, quite important that we have her available for the conversation.
Is it worth having the meeting on the scheduled day and then having a special separate meeting just to discuss that?
Yeah, that's
I'm very much open to that. We can make the 24th meeting just that topic alone. So we'll get it.
One that day, yeah.
Provided Al doesn't, no one mentions the word cell model to Al during that call, we should be able to get it done in in about an hour. So we will leave the April. I think that does anyone have an objection to having 2 meetings next month?
No.
Going in reverse. So we will leave the Wednesday, April 10th meeting as scheduled, and then we'll add in the additional Wednesday, April 24th, specifically with the sole purpose.
If you're having a meeting on May the 4th or whatever, is there any reason to rush a meeting for 24th of April?
I don't believe Eileen can make the meeting. So apparently the first Wednesday of each month is not the best for her.
Okay.
All right.
Okay.
And also, while you're all on the line, just a reminder that I'll be attending the AACR at the end of this month, when we will be talking about this project.
Excellent.
Okay, yeah. Is there anything you need from us to help? Any slides? Anything you can get? Let us know.
I'm finalising the slides at the moment, and I'll send them round hopefully early next week, and see if you think there's anything else. It's only a relatively short talk. It's only 10 minutes, so don't have too much in there.
Yeah, can you give us the information about the talk, like, so that we can all attend? I mean, some of us are going to the AACR meeting too.
Yeah. I mean, it would be great if anyone is, it's on the Tuesday, the 2nd of April, in the afternoon. It's the chemistry, cancer chemistry session.
Okay.
For anyone.
Good, we'll bring tomatoes.
It would be great to meet up with anyone who is there.
Yeah, great. Yeah, it would be good.
So Charles River also making quite a big thing of this. So
excellent.
Keen on, yeah.
Okay. I have an AOB.
Yeah.
So just for gossip more than anything, we had a $1.5 million donation to launch M4ND for neurological disease. So folks are getting excited about what you guys are accomplishing, enough that some donor gave some money to start it in another indication. And recall that M4K, we're getting everyone to work with us, knowing full well that the company is owned by a charity we set up called the Agora Open Science Charity. And to further give people confidence that the charity is above board and will spend any profits, which are probably none, but Michael Morgan has agreed to be one of the governors of it. He was Wellcome Trust's principal lead on the Human Genome Project, and so he's very well respected internationally. And a verbal but not yet written commitment by Els Torili. She's the Director of Access to Medicines for MSF, for Doctors Without Borders in Geneva. So we have a Wellcome Trust person who headed up the Human Genome Project for them, and the Access to Medicine lead for Doctors Without Borders, having agreed to help us be the overseer of the M4K thing. So I think that's it's good endorsement of what you guys are doing, and we have some very good organizations supporting this. So that's it.
That's good. All right. Thanks everyone for excellent update, excellent presentation. Look forward to seeing you in a month's time. Okay?
Yeah.
Take care. Thank you.
Thank you.